guys, so today we're going to talk about a few things. First, the mechanisms of DNA replication, transcription, and the translation of mRNA into proteins. Okay, so there are many key players in DNA replication, so I'm going to talk about what they are and their functions. So first we have helicase, which unwinds the parental double helix at replication forks. This is known as the scissors because it actually unbinds the parental DNA strand. Next we have the single strand binding protein, which binds to and stabilizes the single stranded DNA until it's used as a template. So this prevents the DNA strands from twisting on each other or binding back to each other. And because there is some strain after being unbinded, topoimerase is responsible for relieving overwinding strain ahead of a replication fork by breaking, swiveling, and rejoining DNA strands. Next we have primase, which is responsible for synthesizing RNA primer at a 5' end of a leading strand and at a 5' end of an Okazaki fragment of a lagging strand. Next we have the two polymerases. So the polymerases are responsible for catalyzing synthesis of a new DNA strand by adding nucleotides to the 3' end of an existing chain. So DNA polymerase 3 is responsible for using parental DNA as a template and synthesizing new DNA. It's synthesizing a new DNA strand. By adding nucleotides to a RNA primer of a already existing DNA strand. Next, we have the DNA polymerase 1, which removes RNA nucleotides of a primer from a 5' end and replaces them with DNA nucleotides added to a 3' end of an adjacent fragment. So this removes the RNA. This one retranslates the already added RNA nucleotides and replaces them with DNA nucleotides that we actually have in our DNA. Lastly, we have DNA ligase, which joins the Okazaki fragments of lagging strand on a leading strand and joins the 3' end of DNA that replaces primer to the rest of the leading strand. So this is kind of like the glue. After everything has been, after every function has been has happened, it glues everything back together into a new a DNA strand. Okay, so first thing, you may be wondering what is a leading strand and a lagging strand. So a leading strand is continuously elongated in a mandatory 5' to 3' direction. And so a lagging strand, in order to elongate the other new strand of DNA in a mandatory 5' to 3' direction, DNA polymerase must work alongside the other template strand in the direction away from the replication fork. So this is the DNA polymerase has to work in the opposite direction of the leading strand. And in contrast to the leading strand, which elongates continuously, the lagging strand is synthesized discontinuously in a series of segments. So these segments are known as Okazaki fragments. Okay, so now let's actually get into the actual process of DNA replication. So first, after the RNA primer is made, DNA polymerase 3 starts to synthesize the leading strand. Next, the leading strand is elongated continuously in the 5' and to 3' direction as the fork progresses. So in the synthesizing of a lagging strand, first we have the primase joins RNA nucleotides to a primer. The DNA polymerase 3 adds DNA nucleotides to the primer, forming one Okazaki fragment. So after reaching the next RNA primer to the right, DNA polymerase 3 detaches itself. And the fragment 2, after fragment 2 is primed, DNA polymerase 3 then adds DNA nucleotides, detaching when it reaches the fragment 1 primer. And then after this, DNA polymerase 1 replaces the RNA with DNA, and it adds nucleotides to the 3' end of a fragment 1 and later fragment 2. So after all this, DNA ligase forms a bond between the newest DNA and the segment of fragment 1. And the lagging strand is now in its own complete section. Okay, so with that said, now we're going to move on to transcription. So transcription is the synthesis of RNA using information in the DNA. Two nucleotides are written in different forms of the same language, and the information is simply transcribed or rewritten from DNA to RNA. So just as DNA serves as a template for assembling a complementary sequence of RNA nucleotides, for a protein coding gene, the resulting RNA molecule is a faithful transcription of the gene's protein budding instructions. So this type of RNA molecule is called the mRNA or messenger RNA because it carries a genetic message from the DNA of a protein synthesizing machinery of the cell. So next we have translation. So translation is the synthesis of a polypeptide using the information of the RNA. During this stage, there's a change in language, so the cell must translate the nucleotide sequence of a mRNA molecule into the amino acid sequence of a polypeptide. The sites of translation are ribosomes, and so because of a lack of compartmentalization, this allows transcription of an mRNA to begin while the translation of an mRNA to begin while the transcription is still in progress. So the presence of a nuclear envelope separates transcription from translation in space and time. But so while a transcription occurs in the nucleus, mRNA must be transported to the cytoplasm for translation. And also, I hope you guys learned something about this. I mean, it might be a little bit confusing, but I hope you're able to at least gather the basics. So thank you for watching. Bye.